All right, so we were talking about the immunology, and in that the star of the day is neutrophil, and we were talking about neutrophil. This is still the neutrophil lecture. It's just broken up into two pieces. Anyways, so last we left this in our last lecture, if you heard, the player who needs to come into this stage now is the neutrophil. So neutrophil needs to make its entry here, come in here and start doing its action. So we are all waiting for the neutrophil to arrive. So let's see what happens. What we have done is that histamine, thrombin, and normally leukotrienes, bradykinins, these are all tissue breakdown products, or the coagulation system products. These allow these cells to become contracted. Similarly, interleukin-1 and tumor necrosis factor, which come over here, they have certain very interesting functions to do as well. So we are going there now. So first of all, let's understand what does this do. So know this, that histamine and thrombin, leukotriene and bradykinin, leukotriene B4 and bradykinin, could be bradykinin too, but anyways, bradykinin, these cause the endothelial cells to shrink, contract, and then allow the various substances which would be moved from the blood vessel to the tissue. Now, an important thing, very important thing from a USMLE point of view and from a doctor's point of view, this blood vessel which you are seeing here is a venule. It's not a capillary. Don't say it is a capillary. It's a venule. So these are the veins which have gotten endothelial cells, and these cells are contracting. So remember, there is a basement membrane here. So this really is the venule which is going through this. So normally the inflammation starts or appears on the venular ends. Soon it's going to spread everywhere, so you would not be able to differentiate, but that is how it happens. This is important for us to know, for doctors to know, because there is a basement membrane and some tissue which needs to be broken before the things can move into this tissue here. So we'll see if neutrophil is capable of breaking this barrier and coming into the tissue. So once again, focus here. These are those happy cells. Why they're happy? We're talking about them. Okay, what's happening? We've gotten them contracted. We have some gaps appearing between the endothelial cells. That would allow some fluids, proteins to start jumping out of the cell, of the vascular system into the tissue here. What would that do? What will that do? Number one, that would reduce the fluid inside the vessel. Correct? Number two, when the proteins go out, the oncotic pressure outside would increase, which would cause further pull of the fluids outside the tissue. So now this tissue is going to start swelling up. It's going to start becoming red because there is more blood flow in that area. There is more accumulation of the substances in that area. It's swelling up, it's becoming red, it's going to become painful because of many of the arachidonic acid components in there. But the important thing for us to note is that the fluid which is flowing here, the blood which is flowing here, that flow of the blood is going to start reducing. Stasis is happening now. Stasis is happening. What does that mean? So we should look at neutrophil as a jet plane, as a fighter jet. That fighter jet is needed in this area, correct? So remember, if you have seen those fighter jet movies or war events, you see that, I'm going to go and make that over here. So you see that there is a platform present on a ship, right? So let's say this is a ship, right? So there is a runway present on the ship. So the ship is in the ocean, right? And there is a runway on the ship. And remember that the fighter jet planes, they come. So now this is my second test to be able to see if I can make fighter jet plane. Okay, so a fighter jet plane. So the fighter jet plane, that plane is going to come and land here and be stopped here. This is exactly what neutrophil has to do. It has to come into this blood vessel, it has to slow down, then it has to land, it has to start not rolling. I mean, this guy doesn't have to roll, but you can see the wheels of the jet are going to roll and then finally come to a stop. That is the same thing which is going to happen to the neutrophil. That is going to come in the blood vessel. It's going to roll on the margins of the blood vessel, on the wall of the blood vessel, finally stop there and get out of the blood vessel. That is the function, that is the activity we are going to see right now. So, first we have to slow down the plane, the fighter plane. Second, you know that when these planes end up on the runways, so let's say this is the plane. I don't want to spend too much time in continuously drawing the plane. You know, when it puts the wheels down, there are little hooks present on the runway, right? So there are hooks present on the runway which trap the plane or which interact with the plane, and then the plane slowly starts slowing down in speed, and finally, hopefully before the end of this, it comes to a complete halt. So can I say that the wheels of this plane, they are slowly going to roll and finally come to a halt? If these wheels were neutrophil and this runway was blood vessel wall, then what's going to happen is that the neutrophil is going to come, roll, slowly slow down, and finally stop. What we have to understand now is, how does this happen? So let's go back. This is the analogy, this is our example. So let's go back here. So Mr. Neutrophil has come into the, and there are, you know that there are 100 billion neutrophils per liter, right? 2.5 to 7.5 multiplied by 10 raised to the power 9 neutrophils per liter. So there are a lot of neutrophils running around in the blood. So as they enter this blood vessel, which has stasis, which has slow speed of flow of the fluid, these neutrophils will then be pushed on the side. So one, their speed has reduced, and number two, they will be pushed on the side of the blood vessel. Do you know by whom? By the RBCs. So the RBCs which are running, they would kick them, and the neutrophil would start getting pushed towards the wall of the blood vessel. So I can give this example, at least in the Western countries, when there is an ambulance which is going, the cars on the road, they try to go to the side and stop there so that the ambulance has a way to go. Similarly, let's say an RBC is here. That RBC is sort of an ambulance for this example, and because it has to go quickly, it pushes the neutrophil on a side, and the neutrophil gets marginated. I have heard this as well, that the RBCs clump together in the stasis blood, and they make a big mass, and that mass kicks the neutrophils on the side. I really don't know if that is really a good mechanism to say it, but anyways, that could be the reason too. End of the day, this neutrophil comes in contact with these endothelial cells. So stasis or slow down of the blood flow cause the neutrophil to come in contact with the endothelial cell. Now here is a very important thing. These endothelial cells, if I make one endothelial cell over here, I don't want to disturb our picture, but if I make an endothelial cell over here, these endothelial cells have a thing called, oh, let's go here. So if there is an endothelial cell, the cell has, I think you might have heard, the cell has Weibel Palade bodies. Have you heard of those Weibel Palade bodies? What these are little vesicles inside the endothelial cells. And these vesicles contain chemical factors in them. What are the chemical factors which are very important? They, number one, contains P-selectins. The other things which are not relevant here but are important, von Willebrand factor is there. Another thing which is very important here as well is interleukin 8. So von Willebrand factor, remember clotting, interleukin 8, remember interleukin 8 is a chemotactic factor, and Weibel Palade bodies, and the, P-selectins are present here. So what is important for us to understand, I don't know if it is too small to read, but what I've written here is von Willebrand factor, IL-8, and the p-selectins. So what is important for us to understand is, what are these selectins? So here, look this. And these are present in the Weber fluid bodies inside the cytoplasm of the endothelial cells. So check this out. Selectins are actually proteins which are showing on two cells. Let's say their cluster designation is 62. So these are called CD62, P for platelet, E for endothelium, and L for leukocyte. So we say CD62P, CD62E, CD62L. What these are these are P selectins, E selectins, and L selectins. Here is the important thing. L selectin appears on the surface of leukocyte. L for leukocyte. So CD62L or L selectin appears on the surface of the leukocyte. It's a hook. It's a little mechanism to engage. Remember that jet fighter pilot, that the fighter jet, that has to come and slow down and stop. So it's going to engage with certain mechanisms which are going to pull it back. This is endothelial cell that also has the selectins on it. But these selectins are not always showing on the surface. They are usually present in a little packet present inside the cell, and that is a packet we just talked about, that is called Weber fluid body. So the Weber fluid body, that contains the p-selectin, then it contains von Willebrand factor, and then it contains interleukin-8. Right? So now come here for a second. So what happens is that when the histamine and thrombin and leukotriene and bradykinin, when they act on the endothelial cells, what they do is they kick the endothelial cells, machinery kicks these Weber fluid bodies and fuse them with the surface. So when they get fused to the surface, these selectins appear on the surface. So we'll continue and we'll talk about the other selectins in a second.